Hello, good morning. I'm Dr. Aldo Gómez-Benitez. First of all, I apologize for not being live today. I'll be talking about fluctuating asymmetry and differential responses of species and metapopulation, again, anthropization, concretely in Squamates. So, anthropization is the modification of natural habitats due to human activities. It is characterized by a high habitat fragmentation. It causes biodiversity loss at global scales, and Mexico is not exempt of that situation since local extinctions have been already reported in the country. So developmental instability, it's a biological early indicator. It refers to the inability of a genotype to produce the same phenotype under identical environmental conditions. And we can measure that developmental instability through fluctuating asymmetry, which is a random deviation from perfect bilateral symmetry. So it's important to localize biological indicators of an early response to understand how organisms, in this case squamates, are responding to the different conditions in an anthropized habitat. So we can measure that fluctuating asymmetry through geometric morphometrics, which are a set of multivariate statistical response techniques that allow us to study the spatial relationship among anatomical landmarks of interest. So if we employ classic morphometrics or linear morphometrics, we can have some sets of data of the head, in this case of a rattlesnake. But if we instead employ geometric morphometrics, mark some interesting regions in the head of rattlesnake, we will have this set of data, which is a lot of data. So it's a really precise tool to understand morphometrics. Regarding fluctuating asymmetry, there are a lot of responses in different species. Some species have an increased fluctuating asymmetry due to inhabit urban environments. Other species have a more marked fluctuating asymmetry in wild habitats. And of course, there are species that don't even have a response. That means fluctuating asymmetry is equal in wild habitats and in urban habitats. There are two questions that remains regarding fluctuating asymmetry. One is if fluctuating asymmetry could be corrected with growth through a compensatory growth. And if fluctuating asymmetry is a whole body property, that means if you had an asymmetric head, you will have asymmetric also legs, for example. And why to study biological indicators in Mexico? That's because Mexico is one of the mega diverse countries, have 1,000 species of reptiles, 57% of endemism, and that is equal to 8.7% of the world reptiles. Despite that mega diversity, 17% of the reptiles in Mexico are in some category of red list, and 29% are in a risk category according to Mexican laws. So there are constant risk to lost species in Mexico. Then it's necessary to study biological indicators to avoid the effect of anthropization. In this case, we use fluctuating asymmetry. Measuring them using fluctuating geometric morphometrics makes them highly precise conservation tools. And we also can test if there are a differential susceptibility among studied species or among metapopulations due to inhabit anthropized habitats. So our objectives were to evaluate the susceptibility to environmental stress of a squamate community located in an anthropized environment, to evaluate whether the studied squamate species exhibit fluctuating asymmetry when inhabiting an anthropized environment, to assess the spatial pattern of fluctuating asymmetry in a squamate reptile community in an anthropized environment, and to determine which characteristics of anthropization cause the greatest stress in squamate reptiles. So for this, we collect squamates in El Cerrillo, Piedras Blancas, Toluca del Lerdo, state of Mexico, Mexico. The climate in the area is temperate to humid with summer rainfall, and the vegetation is mainly composed of grasslands. And the disturbance in the area is caused by rain-fed agriculture, constant human presence, livestock grazings, and irrigation channels made of concrete that occasionate the kill of some squamates that fall in the canals. So the soil is treated with malathion and some herbicides, just as atrazine and S-metolachlor, that commonly are regulated by laws, but in this site are used without no regulation. We make some photographies of the dorsal part of the head in 5 species. That species were Varicella imbricata, Celopurus grammicus, Crotalus triceriatus, Tapnophis equus, and Tapnophis scalaris. We then mark 22 landmarks in the head of that species that you can see in the slide. And then we use TPS-RELF to conduct a principal component analysis that tell us if we make some mistake in the mark of landmarks or if we have repeated individuals. Then we use a procrusted superimposition, that it's a process that standardizes our landmarks across the individuals collected. For example, if we want to compare the head of that small rattlesnake with that big rattlesnake, we will see difference, of course, because the separation between landmarks will be bigger in the biggest animal. So we need to make some corrections. The first correction is rotate. Then we resize it. And finally, we translate it to occupy the same space. Now we can compare the shapes of both individuals as equivalents, employing softwares just like MorphoBank, for example. And the proper way to study fluctuating asymmetry is to, again, mark our interesting landmarks, identify the left side and the right side, and then we have a composition of landmarks of a real animal. Then we imagine how we build that animal if it is perfectly symmetrical. We again mark our landmarks, identify left and right sides, and then we compare the actual configuration with a symmetric imaginary configuration. To do this, we conduct a procrusted ANOVA, which also let us know how asymmetric are each individual, and we measure it as a Mahalanobis distance, which is our asymmetry index in geometric morphometrics. Also, we conduct a cluster analysis to identify a spatial pattern in fluctuating asymmetry. We use as variables the Mahalanobis distance, our asymmetry index, the latitude and the longitude, employing the method WARTS and Euclidean distances. We also obtain some environmental characteristics to understand which of these characteristics are affecting more to squamates. The characteristic studies were land use change as an NDVI, human presence, chemical pollution, microclimate changes as land surface temperature, and soil moisture index as an index of impermeable surfaces. We also conduct a CART analysis, that is a classification and regression tree analysis, to understand under which conditions squamates are more asymmetrical. Our results reveal that only 3 of the 5 studied species presents fluctuating asymmetry in the study area. That means that species are affected for being there. And that species were Varicella imbricata, Tapnophis equus, and Tapnophis scalaris. Celopurus grammicus and Crotalus triceriatus were not affected by that Anthropization habitat. But why occurred this? Why Crotalus triceriatus and Celopurus grammicus are not affected for the human activities in the area? So, our hypotheses were that maybe Crotalus triceriatus, the rattlesnake, and Celopurus grammicus are more generalist. That means both of them are euthermical, both of them have a generalist diet, and both of them have a geographic distribution, a wide geographic distribution. That it's contrasting with the both Tapnophis and Varicella imbricata that have more characteristics of an specialized species. Another hypothesis is that the habits of our species are causing more or less environmental stress. That is, Crotalus triceriatus and Celoporus grammicus are completely terrestrial, and both Amnophis use water bodies to prey and to feed, and Baricimbricata use also water bodies to escape. We know that agrochemicals are concentrated mostly in water bodies. So if you don't enter the water bodies, you are only retaining or in contact with certain amount of agrochemicals. But if you swim in water bodies near to crops, you will find a lot of high percent of the agrochemicals in the water. So Baricimbricata and both Amnophis are entering in a more direct contact with the agrochemicals than Crotalus triceriatus and Celoporus grammicus. We identify a consistent spatial pattern in fluctuating asymmetry. We identified 3 clusters: the less affected, which is the blue, the moderately affected, which is the yellow, and the highly affected, which is the red one. When we compare Mahalunovis distance between that clusters or metapopulations, we identify that there are differences between the metapopulations, Mahalunovis distance, or of course asymmetry. And we did not find difference in asymmetry between the 3 affected species. So only metapopulations are a factor that could influence the presence of fluctuating asymmetry, or the amount of fluctuating asymmetry. When we compare the environmental characteristics between species and metapopulations, we found that distance from buildings present difference in both in species and metapopulations, and of course an interaction. The moderately affected metapopulation are farthest from buildings, and the highly affected is more near to that building. So there is a component of constant human presence. Regarding distance from crops, we found no difference between species, but difference between metapopulations, with again moderately affected metapopulation being the farthest from the crops. Regarding the NDVI slope, which is the changes in greenish in the area, we found that highly affected and less affected metapopulations were identical. Regarding land surface temperature slope, we found that the less affected metapopulation inhabits in a site that tends to get color when we compare it with moderately and highly affected. And regarding soil moisture index slope, we find no difference between metapopulations, but between species, with Dapnophis eques and Baricimbricata being identical. That was our classification and regression tree for Mahalunovis distance, or for asymmetry. And as you can see, being farthest from crops but nearest to builds occasionate the highest amount of fluctuating asymmetry, that is 3.6. And of course in the slide you can see another conditions to have more or less fluctuating asymmetry. But the part that it's more important for us to conduct that chart, it's to obtain an important value for each variable, which are these. As you can see, NDVI slope was the most important characteristic. That is an indicator of land use change, followed by distance to buildings, an indicator of human presence, and distance to crops. That could be also an indicator of pollution. Land surface temperature and soil moisture index were the less important variables in our study. So as you can see, anthropization includes a lot of characteristics, so we have to study it as a multifactorial phenomenon. And that is. Thank you so much for your attention. That's my email contact. I want to ask you all of your questions. I'm really excited about it. Thanks so much.